There we go. We're recording not live. This is offline. It's Taran Murray's Rate Good podcast number 3 with Mike McPhee.
How's he getting on?
How's he getting on?
It's right good.
The cat's here and Matt's here. You want to be involved, Matt? You want to say anything?
I might hang out a little bit, but I also want to lay down.
Yeah, Matt's hurting. You all should be grateful that you're not in pain, because Matt's in pain. It's not fair. But Mike McPhee, he's a cool guy. He did play bass in a band that was fucking sick called Oceanic, and they have an album. They have 2 albums out. You can find them on iTunes, and I'm sure other places. Mike, I'm sure, will tell me. But,
there's a band camp.
There's a band camp. Find it. Oceanic is the name of the band. They have an album that's self-titled, or was it called Origin?
Well, I think it was called Origin. Yeah, and then that was the first album, and then the second one's Trappist.
Fuck yeah. They're fucking super sweet. Mike here played the bass on those records, and you should totally check him out. But Mike's also used to be a roommate of mine and Matt's, and he's studying. You're like at the forefront of some cancer research at Dalhousie University too.
Trying to be, yeah.
Trying to be. So that's super cool. So we'll probably get into some shit about music, some shit about cancer, and wherever else the conversation goes. So
yeah, maybe we'll see where this goes. Maybe talk about snow leopards a bit more.
Snow leopards, that was a good topic. So what were you saying about you heard about the snow leopards sound?
So I don't know. Glenn was talking. So it's Glenn O'Keefe, who plays guitar in Oceanic. I live with him. He was just talking about snow leopards, and if I remember correctly, he just was going no, and making this noise, and he's like, "That's what a snow leopard sounds like." So I admittedly, when I find something funny, I obsess about it to like no end. So I've been obsessed with snow leopards all day. I just find they're so hilarious. They make this ridiculously loud calling noise.
It's so ironic if that's the case, because they're just one of the most beautiful cats.
Oh yeah, they're gorgeous animals. Unfortunately, I'm under the impression that maybe they're not doing so well in terms of their population. Oh, this is the footage I'm pretty sure I saw.
Let's play it.
So this makes me sad because he's like in a little cage.
Yeah. Maybe he's sad because he's in the cage.
Yeah. Well, I don't think this is the one.
Here comes.
Look at this.
Oh my, the size of those paws.
As you can tell, it's a bit shy.
This guy could have edited this video better.
Yeah.
Who are you?
It's anticlimactic.
Ted and Genoc on YouTube.
Do not follow, unsubscribe.
Yeah, don't. Yeah, edit your videos, man. I'm fucking on that thing to hear the snow leopard, and you got a bunch of bullshit with your
I don't
kids
need the
saying.
preamble.
Yeah, those little kids saying, "I can see you."
Yeah. Okay. So, anyways.
So the snow leopard, that was a fail, unfortunately. But
oh well.
Apparently, Glenn heard the sound of snow leopard. I think on Planet Earth, so I guess that's where he got it.
Alright.
We'll have to watch that and get back to people.
Yeah.
But I feel like I didn't introduce past people on the podcast, so I think I should do that real quick. I introduced Dylan, who was on last, but like Robin here, you grew up in the Mahone Bay area. You're a painter, and you're looking up information. And so she's the lady who's in charge of information. We don't know it if she doesn't know it.
I don't know it if Google doesn't know it.
Yeah. And Anton was here the last 2 times, and he's just a super tight bro from Hants area, Nova Scotia. And him and I have played in bands together, played guitar together. We were in school together for music. And so we've been tight friends ever since. Matt, he's not been around, but he's been around. So
Yeah.
He's not been saying anything, but
He's upstairs.
he's
at least.
upstairs listening.
And making some
Yeah,
music.
you should listen to his music. Plural would be the most recent project, I guess. But yeah, check out Plural. Find it on iTunes, YouTube, I'm sure. Bandcamp, probably. Yeah.
Definitely on Bandcamp. I don't think we have anything on iTunes.
Nothing on iTunes. That's true. I think I looked for you.
Yeah.
Yeah. But another fucking sick musician
Yeah.
around town. But yeah. So fucking, what are you up to lately at like Dalhousie?
So
Anything cool? How are your experiments going?
they're going good, I guess. So basically, I'm part of this this cancer research traineeship program. So I've been part of that for the last year, and I'm now entering the final 8 months of the program. And my project that I've been working on for that period of time is looking at basically how cancer cells are able to survive in conditions where most cells would die. Cancer cells, they tend to be very adaptive to their environments, and so I've just been looking at how cancer cells might be utilizing certain pro-survival pathways to basically evade cell death, because generally when cells get into conditions where they don't like to grow or they can't really grow well, they'll just kill themselves. For example, the particular phenomena that we investigate is how cancer cells can survive when they detach from a basement membrane or other cells. Generally, if you have like a typical cell in your body, say like in the case of the cells we look at, the cells that line the intestine, if an intestinal cell detaches from other cells or its basement membrane, it'll die. It'll undergo a process called anoikis. It's a cell death triggered by detaching and not being able to grow. So it doesn't have an input signal that says, you're attached to something. Cancer cells, malignancy basically occurs when a cancer cell can detach from a basement membrane or other cells, and it's basically like, that's fine, I can still keep growing. So we were studying how lipids being like a type of biomolecule, make up membranes, fats, all sorts of parts of your body. We're looking at how synthesis of membranes by cancer cells differs and might contribute to evading cell death. So for the last year, I've been working on this project, looking at how these lipids might be utilized by cells in what's called autophagy. Basically, this process is used to degrade internal components within a cell. So if a cell detaches from a basement membrane, at times will upregulate this process to help survive. And no one really understands it. So we were thinking that maybe this was happening in the cancer cells we were looking at. But I recently did some experiments that ruled that out. So we're not really sure where we're going with this. I'm working on some other projects that aren't directly related to that. But right now we're kind of back to the drawing board.
So it ruled out the possibility that you could learn what made cancer cells able to survive on their own like that and teach real, like normal cells, healthy cells, how to do that, or
no? It's more like what we found out is that that certain lipids that we were trying to prevent the cell from being able to synthesize did not change whether a cell could use the process called autophagy to help survive. That's what we ruled out. So we haven't even really established in these particular cells that autophagy is a pro-survival. In a lot of cells, cancer cells, it is pro-survival. So basically, what we found is that at the very least, the synthesis of a lipid called phosphatidylcholine, which is a membrane lipid, whether you can synthesize that or not, does not appear to affect autophagy in these cells. So that tells us something, that that this lipid isn't important for autophagy. But what we do know based on other experiments is that this lipid is important for cell survival. If you prevent these cells from being able to synthesize this lipid, cancer cells in particular, they cannot survive in detached conditions. So the overarching theme of this project is to look at how that happens. So looking at autophagy was just one mechanism where we thought it might be contributing to cancer survival.
So autophagy is when it's surviving while detached, right?
And so that autophagy is self-eating, essentially. When the cell eats
when it kills itself.
No, that's cell death.
Okay.
So autophagy is when a cell basically it sequesters internal components. It'd be like if we sequestered a part of our liver and then digests it. Cells can do that.
Remember that ear-to-ear song, Autolysis?
Oh yeah.
So yeah, like this is like
shit.
Autophagy just means like self-eating. What cell death is called is called
fuck yeah.
So cell death is called apoptosis. So autophagy can either be pro-apoptosis or it can actually repress apoptosis in very particular conditions. It's very complex. So there's one part of our lab that actually has shown that autophagy in precancer cells is important for when cells detach, actually triggering the cells to die. It's a good thing. And when cancer cells detach, that autophagy actually has been shown in some cases to promote survival. It's all about the context of the particular process in the cell. But for my project, we're kind of thinking that autophagy isn't related to the pro-survival phenotype that we see when we allow those cancer cells to synthesize a lipid called phosphatidylcholine. So basically, if I were to summarize my project now, it's basically just understanding how phosphatidylcholine contributes to cancer cell survival. So if you grow cancer cells in a media without nutrients that are required for the synthesis of phosphatidylcholine, those cancer cells can't form tumors. So you can actually grow cancer cells in detached conditions. You grow them in these plates with like sort of this gel-like surface that they can't attach to, so these particular cells will form like balls of cells. Non-cancerous cells, you really can't do that. But if you deprive those cells of the amines to synthesize this lipid, they'll die. So we're just trying to understand why that's the case, the mechanism for that. It's all looking at mechanistic things. We're not so much at the translational stage where we're trying to figure out how can we target this for like a potential cancer therapy. We're not there yet.
Okay.
I mean, you could like based off of some of my work, like it looks like there's a particular enzyme in the cancer cells. There's a particular enzyme in the cancer cells that in normal cells that aren't cancerous, it doesn't really appear to be doing a whole lot. In cancer cells, there's a lot of it, but it still doesn't seem to be doing a whole lot. But what we suspect is happening is when those cancer cells detach and they're sort of flowing around in this, like either the blood or they're like just simply in the lumen of your intestine or something like that, we think that the activity of this enzyme that is responsible for phosphatidylcholine biosynthesis, we think that its activity is upregulated and it plays a role in preventing apoptosis or anoikis. Anoikis is a form of apoptosis, which is a form of cell death.
Okay.
So that's sort of.
So it's pretty like basic as fuck and super easy for anyone to understand.
I hope so.
I'm only saying that because it's a joke.
Oh, sorry.
It sounds very complicated.
Sorry,
but
I wish I could.
You're the expert in the field, so like.
Oh gosh.
Okay, so if I'm understanding this correctly, it's that so these cancer cells detach from like the main tumor or the main.
It's not even. It could be the tumor. It could also just be
attached to any organ or.
Yeah, where at the site of where it first became malignant, meaning like able to spread and to metastasize.
And so, like, if it's flowing through your blood, what would be the risk, or would it attach to some other place it winds up eventually, and then start just propagating in that area and cause a tumor?
Precisely. So the sort of one of the biggest problems with cancers is because they can survive detaching, they can spread and metastasize, forming secondary tumors.
So if you find out like what might be causing their survival while detached and stop it, you could stop them from spreading theoretically.
Exactly. So that is the sort of the potential translational and maybe even eventually like a clinical aspect of doing this research. It's the reason why the lab I'm in first started looking into it.
Okay, interesting.
Yeah, basically the whole goal is preventing cancer cells from spreading.
Nice.
You summarized that very well.
Sweet. So what is a lipid and what does that do in a cell, and how is this lipid in particular you're talking about? I mean, I guess that's what you're studying is how this particular one helps the survival, right?
Yeah.
But what would a lipid, by like general umbrella definition, be? Do they occur in cells regularly?
Oh yeah. Lipids are so basically lipids primarily make up the membranes that surround every cell in your body. Okay. Lipids form these sort of bilayer sheets that surround not only the cell, but its internal components. Cells kind of have what are called organelles. They're similar to our organs. They have like functional compartments, and these compartments are membrane bound. Lipids form all these membranes. Lipids are generally either hydrophobic, meaning they don't like to associate with water, or they're polar in that one portion of the lipid is water loving, and then the other part of the lipid, for example, fatty acids. Fatty acids have a long chain of hydrocarbons that the hydrocarbons don't like to interface with the water, but there is a what's called a carboxylic acid end to the fatty acid chain that can associate with the water.